Hey guys, today we're going to talk about Chapter 4, Single Gene Inheritance, and these are the last two sections for the chapter. So Section 4 looks at pedigree analysis, and then just some extra notes from Chapter 5. All right, so you guys ready? All right, let's go. So first let's talk about pedigrees. Pedigree charts depict family relationships and transmission of inherited traits. So it's kind of like a family tree, but we're going to use shapes and symbols and shading in order to track a certain trait through the different generations. Now we mentioned that they have symbols. Squares represent males, XY individuals, and circles represent females, XX individuals. You can see there's an example of one on the bottom right. So there are 3 males in this pedigree. 1, 2, 3. There are 2 females. 1, 2. Horizontal lines indicate parents. So a lot of times when you see them written like this, a lot of times they'll actually be written up or drawn across the center between them. When there's a horizontal line that connects a square and a circle, then that indicates parents. The vertical line going down shows the change in generation from the parents to the offspring. And this elevated horizontal line that the offspring are hanging from, like grapes on a vine, represents the siblings. So here we have a 2-generation pedigree, the parent generation and the first filial generation, F1. Let's see, they have 2 sons and a daughter. Now, a lot of times we'll put them in order from oldest on the left to youngest on the right. So this son is the oldest, the middle child, and the daughter is the youngest. Sometimes if a person is a carrier or a heterozygote for a particular trait, they might show them half shaded. If they are shaded though completely, then that means they have whatever trait we are looking at. So in this pedigree, there are 1, 2, 3 people with the trait that we are tracking. Whatever it is, 3 individuals are expressing it. And then there are 2 that are not shaded, so there are 2 that do not have the trait. So sometimes if it's someone is a carrier or heterozygous, they might draw them as half shaded. But whoever has the phenotype, whoever is expressing the trait that we are looking at, they are always completely shaded. So why do we want to use these? Well, they can reveal the mode of inheritance. So that means it can tell us, is it sex-linked or is it autosomal? Is it dominant or is it recessive? It can also include molecular information. For instance, in our previous session, we talked about the biochemical basis of how recessive and dominant disorders work. So by knowing if it is recessive or dominant, that gives us an idea of the molecular function or lack of function that is going on. We can also figure out if someone is a carrier. So if they maybe aren't expressing it, but they have the allele for it. And it can also tell us if there are input from other genes or the environment by showing us that there's no clear pattern, then that means that something else might be going on. However, it does get tricky to interpret pedigrees because we might be missing information. Now, different ways we can be missing information is if we're not able to talk to everyone and we don't know, exactly the true numbers of who in the family has a trait versus those that don't. Adoption, especially if it's a closed adoption, you might not know the family history of the individual, so it's hard to include them in this family when they're not biologically related. And if you don't know their traits coming in because you don't know their parents, then it's hard to figure out exactly whether or not they can pass on certain traits. Children born out of wedlock, that's not so much an issue for today as it was, further in our history, because a lot of times when some of these were done, they were looking at noble family trees and lineages, and they didn't include children out of wedlock. So in cases where you're looking strictly at the known individuals, and you know that, and you're only looking at children from a given marriage, if there are any children outside of the wedlock, that could interfere. Assisted reproductive technologies such as artificial insemination or in vitro fertilization, only because it's not common, but there are cases where, oops, they used the wrong sperm to fertilize an egg, and now the genetic information might be different than you expect. Or lack of penetrance. Remember, penetrance is simply looking at how much of that phenotype is being shown. For instance, polydactyl, which is more than 5 fingers, has a wide range that it can be shown. It has a huge penetrance. So you can have like multiple fingers. You might just have like a little tiny nubbin sticking off the side of your thumb. Or for whatever reason, maybe you have the gene for it, but for whatever reason, it's not being shown. So you might not know you have this particular trait because its expression is so minimal, you don't even recognize it. Now, if we look at the trait down here, or the pedigree down here, we have 3 generations, 1, 2, 3. And in this, we see that 5 people have the trait. 1, 2, 3, 4, 5. Sorry. Now, given how only 5 out of all these individuals have it, it's likely to be a recessive trait. It's likely to be recessive. So what we notice here, we have female, female, male, female, female, all that have it. So just by looking at this, I can start making certain observations about it. So there's some more pictures. Here's another one. We have 4 generations here. We have 1, 2, 3, 4. And we notice something interesting here. Out of the individuals, 4 males are showing the trait, only 1 female. They also showed us carriers. So we have 4 females that are half shaded. That means that they carry the allele, but they are not expressing the trait. Now this actually tells me a lot. This is telling me that this trait is sex-linked recessive. I can tell all that just by looking at this picture. We see a clear difference in males versus females on who's expressing it. 4 males and only 1 female are showing the trait. And we are also noticing that there are no male carriers. All of the carriers are females. There's no male carriers at all. So that means that this is not just sex-linked, it is X-linked recessive. I know it's recessive because the fact that we have carriers mean that they have the allele without showing it. And the fact that only females are carriers and that more males are affected than females tells me that it is X-linked recessive. Here's another one. We have 4 generations, and I can tell, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 individuals have this trait. Now, just by looking at this, it looks fairly close in male and female distribution, so it is likely to be autosomal. And I can also tell right off the bat, this has to be recessive. There's no way it can be dominant. The reason for that is here. If the shaded ones represented a dominant trait, that means any that is non-shaded would only have recessive alleles. Can these 2 recessive parents pass on dominant alleles? The answer is no, Because to show a recessive trait, you have to have only recessive alleles. So you have 2 recessive alleles from mom, 2 recessive alleles from dad, no dominant alleles to pass on. So the fact that these parents have 2 kids that are affected when they themselves are not tells me they have to be heterozygous. So they have 1 dominant allele and 1 recessive allele. And their first son and second daughter each got the recessive allele from mom and dad, allowing them to show whatever trait we're looking at. Also here, 3rd generation individuals 1 and 2. They are recessive. They can only pass on recessive alleles, so every single one of their children is recessive. All right, a little bit of autosomal recessive information. Some of this we already talked about. We mentioned how it can skip generations because if someone is heterozygous, they're showing the dominant trait, but they have that hidden recessive allele. And autosomal means it can affect both males and females equally. As a review, often refers to loss of a function of the protein. Blood relatives that have children together have higher risk of children with rare blood disorder. And that's because of consanguinity, which is shared blood or incest. That's why you see a lot of recessive traits, a lot of recessive disorders appearing in certain royal family lines in Europe and all that, because to keep the bloodlines pure, royal families would marry within themselves. So that's why you see a higher incidence rate of certain rare disorders in these families now because of the incest that went on, many decades or many generations ago. Here's some examples of certain conditions. We have Pku Phenol Ketonuria, which is a loss of enzyme condition. And what it does is that it can actually cause brain damage. So there's a loss of a particular enzyme, and the lack of that enzyme affects the brain, not just how it functions, but it actually damages the cells in it if it's not treated. Tay-Sachs causes a buildup of lipids in the cells that surround the neurons, causing them to swell up and start choking the neurons. It is a lysosome disorder. We talked about this back in chapter 2. Cystic fibrosis is caused by faulty sodium channels. Albinism, like this young lady in the bottom left, is a lack of pigment. And sickle cell disease is when red blood cells, because of a mutation in the hemoglobin, affects the shape of the red blood cells, and they end up being longer and more curved shape. All right, and autosomal dominant information. Again, quick review. Autosomal dominant traits generally do not skip generations, because when you skip a generation, it means that it's not being seen. If you're not seeing a dominant trait, it's because there is no dominant allele to see, which means if there's no dominant allele, you can't pass one along. And it's autosomal, so it'll affect both males and females. They result from production of abnormal protein that either gains a new job or a new function, or it interferes with the functions of other proteins. So the protein is there and it's doing something, it's just not doing the right thing. Here are some autosomal dominant conditions. We have achondroplasia, like Peter Dinklage here on the left-hand side, also known as dwarfism. Polydactyl, which is when there are extra fingers or toes. For instance, this individual here who has 1, 2, 3, 4, 5, 6, 7, 8 digits. 8 digits on each foot. Huntington's disease, which affects the brain, the personality. It also affects motor function and cognitive ability. Lactose intolerance is the inability to digest lactose, which is a sugar found in milk. All right. Since we're at the end of the chapter, we'll talk about some of the additional resources we have. So first are the Science in Pajamas, the ones that I make and put on YouTube. We have one that talks about Mendel and just kind of an introduction to genetics. We have one on chromosomal separation, so looking at the law of segregation and whatnot. We have one on how to set up, fill in, and analyze Punnett squares. We have one on how to set up, fill in, and analyze dihybrid crosses. And we also have one on how to interpret and analyze pedigrees along with karyotypes, which I know we haven't really gotten a whole lot into, but we have mentioned them. The video is mostly about pedigrees anyways. All right, and other video resources include the Amoeba Sisters, Alleles and Genes. I love the Amoeba Sisters. They do a great job of breaking stuff down. Monohybrid parents and Punnett square guinea pigs. Amoeba Sisters dihybrid and 2 trait crosses. Ripley, I'm almost done, Ripley. Amoeba Sisters pedigrees. Let's see. Biology professor Mendel's law of segregation. Biology professor Mendel's law of independent assortment. And Jared's bio tutorials, segregation versus independent assortment. So these, along with the Science in Pajama video, should help you gain better understanding if you still have questions or confused. You can also hit me up in Google Classroom or through email with questions. I will post a link for this presentation in the video description. So in the meantime, you guys, stay awesome, stay curious, and most importantly, stay safe and healthy. All right, you guys. Bye-bye.